Yes, good evening to everyone. So, welcome to day 3 session, the day 13 session on bioinformatics approaches for drug discovery. So, we discuss various methods of computer aided drug design on today's session. Before stepping it on to today's session contents, okay, that is on drug discovery and applications, drug design and its types. We discuss the methods in drug design, virtual screening, molecular talking. So we can recap the last session topics. So as usual, what are the last session topics we have discussed? So come up with your answers. So what are the last session topics we have discussed? That we discussed on microarray. Other than that, just microarray alone you have remembered. MATLAB. Like in MATLAB, we discussed on Simulink, isn't it? No, Symbiology. We have gone through Symbiology tool. Other than the Symbiology tool, we also have Bioinformatics toolbox. Like if you want to work on uh, the sequence alignment, protein-protein interaction networks, so we can use even this kind of a toolbox. And if you see further, we discussed on the steps in microarrays. Other than that, we discussed on cellular computing. Inside the cellular computing, we discussed on DNA computing and biomolecular computing. Okay, so these are the information we discussed in the last session. Today, we have to discuss a lot of information in this. This is the version, the bioinformatics approaches for drug discovery. Let me go ahead on to today's session topics. So first slide. So how researchers discover the drug for a disease? Generally, for discovering a drug, okay, one should have two important terms. The most important term is the target receptor. Is the target receptor of a disease. Okay, so we can either call it as a target or we can also call it as a receptor. This is one important term. The next important term, what we can find in the drug design is ligand. So ligand is generally actually a small molecule, okay, or it can also be a macromolecule, but it's a small molecule when compared to target molecule. So ligand is generally a drug or it can be inhibitor. Okay, so these are the terms we use for a ligand. So ligand and receptor. Okay, so if only if you have this information for a disease, we should have a target receptor, and to know about the drug, so we should have the 3D structure. Okay, the 3D structure of ligand as well as the 3D structure of the receptor are both are important to come up with a drug discovery. So here you can able to see whatever you can see is a ligand. It is docking in the surface of the receptor. Okay, this is what the scientist is doing now. And drug discovery and applications. Earlier, for computational studies, before a drug is coming to a market, okay, it has took around the computational studies around it took around 4 years. But nowadays, what has happened, just because of this revolution, just because of the revolution of supercomputing. So even if you see when we face the COVID-19 pandemic situation, they have taken just not more than 2 months. Within 2 months, they have repurposed. They have screened the repurposing drugs. Okay, so one scientist have done screening of repurposing drugs from 1.3 billion compounds from zinc 20 database. At that time, they had a zinc 50. Okay, so similarly, whatever databases we have, PubChem for screening the compounds for SARS-CoV-2 for targeting SARS-CoV-2 spike glycoprotein. It took just a few months alone. Okay, and after that, whatever the drug screen, we have to take it to the lead development. Okay, and then we have to try with the preclinical studies. You know, when we faced the pandemic situation with the SARS-CoV-2 virus, okay, so we know that the The world has declared the pandemic in March 2019. But when did we receive the kind of a vaccine? If you see the vaccine in the end of year 2020, okay, if you know the rich countries like US also got the vaccines in September 2020 itself. So it has took around 1.5 year, isn't it? Within 1.5 years, so the computational studies are done. Okay, these are actually computational studies, including lead development, comes under computational studies, and even other than the lead design, if you go develop a drug, of course, we used even in vitro studies for development of the drug. Okay, so whatever tried, okay, after this preclinical studies, we should at least try for minimum 6 months. If you want to work on a drug or a vaccine, obviously minimum if we work for 6 months, we can get a result, isn't it? Very rapid kind of a research happened, and within 1.5 year, we can able to see around 89 vaccines. 89 vaccines came during that period. But whatever vaccines we received, Covishield, Covaxin, okay, we got in 2021. Okay, so the based on our Indian population, so many kind of vaccines, I mean, so many units of vaccines are getting manufactured, and so we get delayed. But almost in this 1.5 year, we can just see 8 9 types of vaccine like Covishield, Covax for SARS-CoV-2. So nowadays, you see this kind of a chart actually taken 15 years before, but now because of rapid computing, parallel computing, whatever we discussed in cellular computing, okay, so we find this drug discovery is possible. The computational drug discovery can able to make it within few months, and then the rest we take it for preclinical development is checking the effect of a drug with animals. Generally, in India, we have permission to work on only small animals, isn't it? Even the SVC has animal house facility, so one can able to study the effect of drug with mice, rat, hamsters, and then they can also try with rabbit. Okay, in our institution, even my PhD scholar have worked on mice recently. And after working on this preclinical, one has to at least check with this phase 1 to phase 3. Not only this has took around 1.5 years. This has took around 9 months, and the remaining 9 months they have replaced for phase 1, phase 2, and also phase 3 trials. So very sharply within 1.5 year they come up with this kind of a vaccine also. Okay, got it. So there is a rapid development in design and development. And if you see this drug design, the drug design classified into majorly two types. The first is the traditional drug design. So before 1970s, how the patients are treated, we use this kind of a pestle and mortar, isn't it? We use the plant materials, grind some 2 or 3 plant sources. We get a kind of a form of a drug. We prepare in the form of a churnam, or we prepare in the form of a paste. Nowadays, even we have Ayush, Ayurveda, Yoga, Unani, Siddha, Homeopathy. All these things also majorly uses plant as the source for the drugs, isn't it, for treatment of various diseases. So it's a random kind of natural ligands we can get that. Okay, if you take to relieve from cold, the best tree is neem leaves. If you use neem leaves, it is best used for treatment of cold and cough. But if you see this neem leaves, we make it more than 200 compounds. We make it more than 200 compounds. Not all the 200 compounds can be used to cure cold. Then how many can able to cure? Maybe around 50 compounds. And the remaining 100 compounds are no use, and maybe the 50 compounds can give even side effects. Okay, then we have to remove this 150. We have to take only the 50. Even from the 50, which is more potential to treat cold, we can able to isolate that kind of a compounds. If you make it, then obviously the treatment is easier for us, and it is very rapid, and even we can fix the doses. So this is quite easier for treatment. Okay, so the traditional drug design care is overruled by structure based drug design. In fact, we need a 3D structure of a ligand. Okay, and also we need a 3D structure of the receptor. Even if you have the 3D structure of a ligand and receptor, then we can do some docking or any other pharmacophore mapping. We can use any of these studies and we can come up with a kind of a precision based drug design. Okay, like whether the drug is bound with the target receptor or not. Okay, this information can be determined easily. So this is the second based of drug design. Nowadays we are living in this structure based drug design and revolutioning drug discovery in silico. If you see the importance of this drug design, okay, one such example you can see is Jolgensma. These statistics I have taken in around 2018 or 19. In 2019, I have taken statistics. So, there is a medicine called Zolgensma. It is actually known as Gene AB Parovec. Okay, so this is actually used for treatment of muscular atrophy, that is spinal muscular atrophy. This is generally occurring in 1.5 year baby. Okay, 1.5 year child can suffer from this spinal muscular atrophy, where if the child, up to 1.5 year, the child don't show any symptoms. But after 1.5 year, the child is mentally affected. It's very difficult to see after the 1.5 year. Okay, so the major response is we have to target this human SMN protein, and for this, one can able to go with this Jolgensma, and this is like a kind of a shot. A single shot is around 5.5 ml, and the expense is around 18 crores. Nowadays, it comes around even 24 crores. Okay, the single shot medicine. So how it is targeting? This is actually gene-based drug delivery. This is like a gene-based drug delivery. For a single shot is not sufficient. This is not sufficient for treatment. One should also go with at minimum 3 shots. Okay, so this is the information. 1 minute. The participants can join on time. Okay, let me go ahead with the presentation. Yeah. So now we understand the importance of drug design. Just as this drug is actually manufactured by Novartis, the only one company which is getting a medicine for this spinal muscular atrophy. Now, let me take you to the computational methods in drug design. So, we have drug likeness prediction, ADMET prediction. Actually, for taking this drug design, I am taking courses like artificial intelligence and machine learning approaches in drug and vaccine design. So this is also a 15 days course. Actually, before this course, I took this course, and for this course, I got around 33 institutions. I got participants from 33 different institutions. Okay, and even when I taken for a different company, 250 participants. That's the maximum I can able to admit. Okay, so 250 participants have joined, and just because many participants want to join this course, we had two different batches. Okay, lot of scope is there in A and ML approaches in drug and vaccine design. This I'll plan to conduct on December 2025. I'll be conducting these courses. Okay, A and ML approaches of drug and vaccine. So in detail, I'll be explaining you the hands-on session with these kind of a software tools. If possible, you can also join. Okay, that's also a 15 days course. So only the basics I can able to come across, and at least one tool I can able to teach. So first we can see about this drug likeness prediction. The drug likeness prediction, why we have to use this drug likeness prediction? Like when I explained you about the types of drug design, a traditional based drug design, okay, we take numerous compounds, and not all the numerous compounds can able to cure a disease. Okay, so only thing is that we have to screen the compounds, and we have to take the best compounds. That's kind of a screening we can also do with this drug likeness. Like if you want to check whether a compound behaves like a drug or not, that is checked with this drug likeness prediction. So this can be done with moles of mole inspiration. So now first take you to the drug likeness prediction. So what do you mean by drug likeness prediction? So the drug likeness prediction can be done with the online databases itself. So I'll be taking you for illustration of two different drug likeness filters. I mean, based on drug likeness filters as well as based on ADM. You know, the pharma students know about ADM. ADME is nothing but absorption, distribution, metabolism, and excretion. So this ADME is a pharmacokinetic property, isn't it? So in Swiss area, there is a website called swissadme.ch where you can able to see a window here. It's a kind of a JavaScript kind of a chemoxan window. One can able to draw the molecule and you can able to click run to find the ADME properties as well as to find the drug likeness properties. So, but there is also one more option for us to enter the SMILES format. SMILES is 1D format of chemical compound. This is a 1D format. So to get the SMILES format, we can able to get canonical SMILES from a database called NCBI PubChem. There is a database called NCBI PubChem. Inside that, we can able to download a 1D format of a chemical compound. For example, if I want to download aspirin, okay, there is also a keyword called aspirin. Either you can type the drug name, compound name, or even receptor name. Here, EGFR is epidermal growth factor receptor, or also we can give disease name like COVID-19. Give any of these keywords to find a related kind of drugs. Like if you give a kind of a disease, what are the possible repurposing drugs we can use it for treatment of disease? So around nearly 1,700 to 1,800 compounds can be generated if you type the COVID-19. But if you want to type a specific drug alone, like an aspirin, okay, I'm just clicking inside, taking it inside. Now I want to take just a smiles format of the aspirin. So to get the smiles format, click the structures. Okay, this is a 2D structure information. If you want to download the 2D structure information, download coordinates STF. Okay, this is the format you can download. Or if you want to download 3D, so this is the STF structure data format. That's the expansion structure data format. You can download this and you can use it for any kind of computational studies. So there is also a format called SMILES, which is a single D format of chemical compound. Whatever aspirin we got, so that this is the 1D format for the aspirin compound. Just like that, I copied the canonical smiles and pasted in this window. We got this run button is enabled. Click this run. So what is going to happen, So this Swiss ADME is going to predict us ADME properties as well as drug likeness properties. What are the drug likeness properties you can able to see in this window? The first drug likeness property what we can see is the molecular weight. So actually, if you see the drug likeness filters, you can see Lipinski filter. The Lipinski filter also called as Pfizer filter. So we call this Lipinski rule of 5. According to the Lipinski rule of 5, the molecular weight should be less than or equals 500 and partition coefficient should be less than or equals to 4.15 and the hydrogen bond acceptor should be less than or equals 10 and hydrogen bond donor should be less than or equals 5. Okay, this is the meaning of these four different properties. Actually, these four properties are included in Lipinski filter. Okay, now let me check about the molecular weight of this compound aspirin. Aspirin 180 is less than 500, so this property is satisfying. And the second property, whatever I'm going to check is the hydrogen bond acceptor should be less than 10. Again, it is satisfying. Hydrogen bond donor should be less than 5. Yes, again it is satisfying. It is mentioned that this should be less than or equals 10 and here also less than or equals 5. So three properties are satisfied. According to the Lipinski rule, If any three out of four properties satisfies, then we can call it as a drug. So now just three properties alone satisfied. Go for the fourth property. The fourth property is the partition coefficient. The partition coefficient, whatever you find here, that should be less than or equals 4.15. We got 1.19. Just because we have 1.19 which is less than 4.15, so all the four properties are satisfied. So according to Lipinski rule of 5, there is no such violation. So zero violation, it shows the drug likeness properties. We also have other drug likeness properties like goose filter, where the properties are changed. There in Lipinski, the molecular weight should be less than or equals to 500. But here, there is a range for the drug likeness filter, 160 to 480. Fortunately, we have in the 180 is between the 160 to 480. So first property is satisfying. W log P. Instead of X log P3, better go with the W log P. 1.31. It is like between minus zero point 0.4 to 5.6. It is also satisfying. Then we have to go with the MR. What is this MR? Actually, molecular refractivity is what MR. 44.9. It is lying between this 40 to 130. So this also getting satisfied, and similarly atoms. So like this, you see almost all the filters, Lipinski, Goose, Weber, Egan, Mugi, all these four filters on only in Mugi we find one violation. The molecular weight should be uh, between 200 to 600, but we have less than 200. So we have only one violation in Mugi filter, but whereas other filters don't have any violation. So this is how we can understand from the drug likeness properties. This is of course a filter for filtering numerous compounds. To come up with the best compound which can act like a drug, then better we try with this kind of a drug likeness filter. Now let me go with the next thing, ADME. ADME absorption. So for absorption, we can check with this gastrointestinal absorption. Generally, for a patients, we suggest to take oral route of drug delivery. If and if you have a good absorption, then it can able to reach the system. So GI absorption of aspirin is high, which is desired. So this is of course a desired property. The next thing is related to the distribution. I told you in ADME, A stands for absorption and D stands for distribution, which we can check with the BBB. BBB stands for blood-brain barrier permeant. Blood-brain barrier. So how come we can able to determine this blood-brain barrier? According to the chemistry, we can able to check through octanol water partition coefficient. Like if you want to decide whether a drug is lipophilic or hydrophilic, better try with this kind of blood-brain barrier where the brain has more lipophilic. Okay, and the blood is not that much. Okay, so because the blood is carrying the water, the maximum composition of the blood is water. So to check the kind, okay, if it is if the blood-brain barrier permeant is high, is yes, this kind of a drug is also lipophilic in nature. So it can also affect the central nervous system. It should not. Okay, if you are treating central nervous disorders, no problem. We have to get a desired property assess. But whatever drug we take, it can also reach us these. That's what you can understand from this drug, this information. And the next is the metabolism. The liver has plenty of cytochrome P450 enzymes, which take care of metabolism of drugs also. And whether you want to check whether this act like an inhibitor or not. So from this, you can able to find this information. And finally, excretion. The excretion can be done even through skin. Of course, the major organ for excretion is kidney. No doubt in that. Unutilized drug is excreted in the kidney, but even a part of the drug can also be excreted through skin permeation. Okay, so that information also can be find. So of course you can able to see the ADME properties here. That's what we call as a pharmacokinetics. These are also a kind of a filter for compounds. If you have plenty of compound to screen the best compound, one can try with this kind of a filter, including this filter, drug likeness filter. This is one filter. This is second filters. Okay, so this is something about what we are discussing in computer-aided drug design. Okay, we have completed till now about how to go with the drug likeness prediction. This can also be done with Swiss area. That's what I explained you recently. So Swiss ARME can also be used for drug likeness prediction. We have nearly four filters in Swiss ARME. Other than that, we have gone to the ARME property prediction through Swiss ARME, a single tool. And the third one is QSAR. QSAR stands for quantitative structure activity relationship. This is a ligand-based drug design. If you have a 3D structure of a ligand, that is sufficient enough to design to screen the ligands. And pharmacophore modeling, this is actually a ligand as well as structure based. Ligand as well as structure based. What do you mean by this? If you have a 3D structure of the ligand, that is sufficient enough to screen the drugs. And according to the structure based, if only if you have a 3D structure of a ligand as well as a 3D structure of a receptor, then only you can go with this. So you have two types of drug design in pharmacophore modeling. So this you can able to try with PharmaGist and PharmMapper. These are the freeware tools. And similarly for QSCR prediction, one can try with this Galaxy. DSQSCR, Discovery Studio QSCR is a commercial. And molecular modeling, we already discussed in the previous session how to go with homology modeling through Swiss Modeler and how to go with the threading concept using ITSL. Okay, so this comes under molecular modeling of macromolecules. For modeling the targets, we generally use these kind of servers, Swiss Modeler as well as ITSL, online servers. Molecular talking. So molecular talking, this is the best tool, of course, I have tried. Okay, Discovery Studio Ligand Fit, but this is a commercial one. Not everyone can afford this. So the best thing what you can try is manual autodoc, manual docking using a freeware, free open source software called Autodoc. Okay, today I'll be teaching you how to practice Autodoc 4.0. And invest talking through asset is of course a free online tool. And molecular dynamics, like if you check the binding of a ligand with a receptor, no, there are two important terms in molecular docking. One is ligand, the other is the receptor. The ligand is a drug, and receptor is a protein target of a disease. So, if a molecular docking is used to check the binding affinity of a ligand or drug with target receptor, the molecular dynamics is used to check the same binding of ligand and receptor over a period of time. Over a period of time, generally we use in nanoseconds. Whether the ligand is bound with the receptor, the binding affinity is good over a period of 500 nanoseconds, 200 nanoseconds. That's what we try in molecular dynamics simulation. So we can use a freeware called Promax to do MD simulation, and this is a commercial software, NAMD. And we also use the read across approach to determine the toxic properties of the compound. This is a freeware, free online tool. You just need a smiles format to input in the toxic. Obviously, you can get the toxicity parameters like for each and every organ, whether it is a cardiotoxic or it is a nephrotoxic, okay, neurotoxic. All this information we can get just by pasting the smiles format. And the gene expression profiling and biomarker step. You remember? Even yesterday I was explaining about the cytoscape. Okay, I also sent you various papers, published papers on how I use the cytoscape. All this information is pertaining to the gene expression profiling and biomarkers development. This is what a recent kind of a uh, development in the computer-aided drug design. On virtual screening, we are high throughput screening using in Autodoc itself. There is one tool called PYRX. PYRX stands for Python RX program. Python RX programming. So this is used actually for screening multiple drugs with their target receptor. Here in molecular talking, we just talk a ligand and receptor. But whereas in virtual screening, you talk multiple ligands with a single target receptor. And if you come across the 12th machine learning techniques, if you want to screen the compounds based on docking or any other kind of a property, okay, for inactive compounds, actually whatever active compounds are used as a drugs at present, but there are also inactives and yet to synthesize. Even these compounds whatever present in databases can also be screened based on the machine learning techniques. Okay, deep miner Python programs are used. So now let me take you something about the virtual screening. What do you mean by virtual screening? I just now mentioned screening multiple ligands with a target receptor. Okay, this kind of a virtual screening can also be done through ligand-based methods or structure-based methods. On structure-based methods, we use the protein ligand docking. And ligand-based methods, we can go with similarity searching, pharmacophore mapping, machine learning methods. So this is something about virtual screening. So ligand-based virtual screening methods can be a pharmacophore modeling, QACR, and structure-based is a molecular docking, AMD simulation. Just now I explained. Here you just check the binding of ligand and receptor in docking. But in MD simulation, over a period of time, we check the binding affinity of ligand with receptor. Hybrid based, that is, you can work either with the 3D structure of a ligand, or even you can work with both the 3D structure of a ligand and receptor. That's what we call it as hybrid based. Even pharmacophore based docking is a best example for hybrid based. It works either like ligand based as well as it works also as a structure based. Now let me come across molecular docking. So up to present, do you have any doubts? Yes or no? Yes, participants. I think we have lot of information. I'm speeding up. Huh? But this is something more pertaining to what you are studying at present, isn't it? Is it okay? Yes, good. So one participant has. How about the others? Okay, good. Now let me go ahead with the BPT. I don't think we should waste time. Molecular docking. Molecular docking is the process that involves placing molecules in appropriate configuration to interact with the receptor. See, whatever we are seeing here, this is an immunology. What kind of a branch is this? Immunology, isn't it? One day in immunology, we can see this antigen antibody. So if antigen is a small molecule, okay, this is our ligand now. And if antibody is a larger molecule, it is an antibody. Sometimes also the antigen might be large and the antibody is also small. But what is a large molecule, large size, that we consider as a receptor. So in immunology, you find this antigen antibody interaction. And what is this? Next to this, you can see enzymology. Enzymology only we can able to see this enzyme and substrate reaction. You know that enzyme can react with a substrate in a particular concentration, which is a reversible reaction, which forms an enzyme substrate intermediate complex. And irreversible reaction of this enzyme substrate can release enzyme and product. Okay, so this is what we study in kinetics, enzymology, where you can see the substrate is binding in the active sites of the enzymatic protein. There are two types of technical terms. One is active sites, the other is the binding sites. Generally, if you see this enzymes, we use a term called active sites instead of binding sites. But for the remaining proteins, we call simply as binding sites. Okay. Now let me go to this third subject. This is something like transport and molecule. The best example, this also comes under cell signaling, and this is an example you can take is hemoglobin. Know about the hemoglobin, what molecule is carried in generally. So you can able to see oxygen is carried inside during the inhalation, and carbon dioxide is exhaled, isn't it, in our human body? So hemoglobin is going to take these two gases. Because of the gas deficient in the plant, in the protein, the hemoglobin protein, you can find if the oxygen is dissolving in the hemoglobin, it is going to form oxyhemoglobin, and during the exhalation, the carboxyhemoglobin, it is going to convert as carboxyhemoglobin. So obviously, the gaseous molecule are taken uh, through the transport. Okay, that's what you can understand. Now, if you see this, this is a signal transaction. This kind of area is a signal transduction. Of course, this signal transduction we can also see in major thing in immunology, like IL-2 ligand. There is IL-2 ligand bind with IL-2 receptor, and we also discussed something like insulin ligand is going to have an insulin receptor. So, what most of the immune molecules bind have a ligand and receptor. The ligand is also a protein, receptor is also a protein, but ligand is a small protein and receptor is a macromolecule. So whatever areas of biology we study, so these are we call that as a molecular recognition, isn't it? In the types of biology, that's what we call here as a molecular talking in competition part. It is the terminology. The molecular talking is a terminology, and of course here whatever we take a substrate is a ligand here, and receptor is the enzyme. Similarly, here this is a ligand, and this is the signal is the ligand, and receptor is the protein target. Even acetylcholine binding with the neural receptor, best example for signal transduction. How the neurons think, okay, that's an example. And in molecular talking, majorly the talking softwares differ based on the search algorithm and based on the scoring function. The commercial software shows many scoring functions. Freeware shows limited scoring functions. In Autodoc, we get only one kind of a score, that is binding energy. So the binding energy, whatever we get in Autodoc, is in kilocalorie per mole. Okay, that's the unit you can get. So now let me go ahead with this kind of a methodology. How well it can bind with the receptor. That's what we call it as a molecular docking. So here the scientist is saying how well ligand is binding with the receptor in docking. And one thing we should remember in the docking, whether if you want to check whether ligand is bound appropriately in the binding site or not, then we can use the different algorithms. One such algorithm used in discouraged ligand fit is flood fill algorithm. Here, if you just drop a kind of a protein molecule inside a box, and if you can fill it with water, wherever the water is going to accumulate in the protein, on the surface of protein, the surface of protein, okay, where shows hydrophilic binding site, can show the hydrophilic binding site. And if the binding sites are buried inside, we call it as hydrophobic. Also, we call it as lipophilic. These are the two types of binding sites we can able to find. So only we have to try this kind of a simulation, and through this simulation, the binding sites can be determined. So plenty of information is there. Okay, in docking, simulation, pharmacophore mapping, QSAR, all this information you can learn. Okay, during this kind of a courses, A and ML approach. Okay, so these approaches can be better used for I mean, these kind of certificate course can give you better knowledge. Now, first, let me explain you how to do molecular docking using Autodoc. So, I have already installed the Autodoc. Okay, so to install the kind of Autodoc, we should have four different softwares: Autodoc Suite, Chimera, MGL tools, EDU Pymon. So these are the four tools. All these four tools are freewares. Okay, so I just going to type here. Okay, so what are the tools we need for Autodoc Suite, Chimera, okay, MGL tools, and MGL tools and EDU Pymon. Okay, so these are the tools we have to install. All these are freewares, but each and every tool has its own page. We have to go there and we have to download it. So, any doubts? Okay, so after installation, so how we can find the software is installed? The software we can able to identify. Okay, this is a PMV. This is one such icon, Python molecular viewer, and in Autodoc, this blue color, like a small molecule can binding with the macromolecule receptor. So this is a second icon you can find, and the third icon is CADT, and fourth icon is Vision. If you install all these four softwares, we find these four icons. And if you want to open the Autodoc, simply click this blue color icon, Autodoc Tools. Now I'm going to open this Autodoc 4.0. There are so many types of Autodoc. When I'm explaining about computer aided drug design, I was mentioning you like Autodoc 4.0, Autodoc PYRX. We also have Autodoc Vina, covalent Autodoc. Okay, but now I'm going to explain you with Autodoc 4.0. So, actually, Autodoc is not exactly a freeware. It is a commercial one. For first 15 days, you can use it for free. Later, it asks you to register. But for registration, obviously, I have to pay. So instead of going for register, I click remind me later. So if you click the remind me later, we can use this still for molecular talking. So this is the window of Autodoc. So first, for working with Autodoc, we should have files in desktop for easier usage. So I had a folder called Demo 6. I've already prepared a folder called Demo 6. There I have this 5 files. The first file is autodoc4.exe. When you install this Autodoc tools, we can have this in program files. Just copy this autodoc4.exe, autogrid4.exe in a desktop folder. In a desktop, I've created a folder called Demo 6. So after installation of Autodoc, whatever files you find in program files folder of C drive, copy, copy and paste it here. Okay, copy and paste it here in the desktop. Later, I'm just uploading this tutorial. I believe in WhatsApp, where I just sent you the tutorial also. Okay, so and then we have a ligand and protein. For a ligand and protein, what I did is I just downloaded a ligand complex with a protein file. If you download a PDB file, you find these two files matched with each other. I just split the ligand separately and protein separately, and now I'm going to try doing manual autodocking, manual binding using Autodoc. Okay, so Indinavir is the anti-HIV protease drug, and protease is HIV protease. Now I'm going to work a docking of this ligand with this receptor. You know why purposefully I'm using this as a sample files? Because protease is a very small protein molecule, and of course indinamide is also a small ligand. So for doing docking, if you have a small molecules, I can able to show you the result on today's. So I'm just trying with this kind of a sample files. Whatever I am showing you a demo, we can try with your project files also. The same kind of uh, steps you have to follow. Okay. So now let us begin with how to go with manual docking using Autodoc. So first I'm opening the tutorial. Okay, so you can able to see a tutorial. Okay, the totally 6 steps. 6 steps for docking, and the 7th step for analyzing the docking results. So analyzing the docking results, I'm not going to try with the Python molecular viewer. I'm not going to use PyMore, but I'm going to use Discovery Studio, which is very easier for you to analyze the results. So the 7th step alone I'm going to change, and I'm also going to teach you on today itself. Now let me take you to the first step. The first step, before going to the first step, there are preliminary steps we have to make with this Autodoc software. What is a preliminary step? We have to set a path where we have all the input files. Know that in desktop I have a demo 6 folder where I have all these 5 files. So to set the path, click File, Preferences, Set. File Preferences Set. And then go to the demo 6 folder. So go to the demo 6 folder. In the demo 6 folder, okay, you copy the location. At the top, I click and copy the entire location. After that, there is one folder called startup directory. In the startup directory, just copy and paste the entire folder link. Then click make default, click set. Okay, and then click dismiss. Now the path is set. So now we are going to carry stepwise. The first step they're asking you to is click file read molecule. File read molecule. Obviously, we have to select the macro molecule just because we have set the path demo 6 default it is opening in demo 6. Now, out of Indinavir and protease, which is the macro molecule, obviously protease is the macro molecule. So I'm opening this PDB. In Autodoc, one thing you have to remember. So you can download ligand and receptor in any formats, but during the docking, you have to convert both the ligand and receptor in PDBQT format. PDBQT. So PDBQT formats you have to make it even in Autodoc PVRX and Coherent Autodoc as well as Autodoc Phenom. Okay, so these are the default formats Autodoc can take for molecular docking. Okay, now I just open this molecule. This step is completed. The next is add hydrogens and charges. For this, edit hydrogen, add all hydrogens. Edit hydrogens, add all hydrogens. Now we have default as all hydrogens, so click OK. So you can see a confirmation that white color thing or nothing but hydrogen bonds added to all the amino acids. Now let me take you to the next step. The next step is edit add Coleman charges. Edit add Coleman charges. So that is in charges you have to click add Coleman charges. We get a kind of a prompt box. Simply click OK. Default things you can take it in consideration. Now file save write PDB file save write pdb. So here we have in demo 6 we had a protease, but instead of protease I'm going to save it like protein1.pdb. I just renamed the file as protein1.pdb. In this protein1.pdb, the hydrogen bonds hydrogens are added and even charges are added. So this is the latest saving file protein1.pdb, the same demo 6 folder. Click OK. Okay. Now the entire first step is completed. This first step I have completed. Now let me go to the second step, preparing the ligand. Okay. For this ligand input open ligand. Ligand input open ligand. In the open ligand, we don't find any ligands here because default it is in PDBQT format. So if you want to open the ligand, so select all files. Now you can find the ligand Indinavir. So click this Indinavir because the second step is to open the ligand. So click open. Now the ligand is open. Default whatever you get, accept. So I just can able to see a ligand, but still you can see a ligand in front of near the receptor. It doesn't mean that ligand has stopped. What you are placing the ligand in this visualizer, in this Python molecular visualizer. Now let me take you to the next step. Ligand torsion tree detect truth. This is what we have to do. Ligand torsion tree detect truth. So this step is completed at present. Now we have to go with ligand output save as PDBQT. Here only you are saving ligand output save as PDBQT. Now we are taking first step, okay, with the ligand. So while saving this ligand as PDBQT, I am saving like ligand1 dot PDBQT. So now this ligand is in the form of PDBQT format. Click save. So now I have completed the second step completely. So where I just had the ligand alone now in PDBQT format. Still we didn't convert the protein as PDBQT format, which will be making steps in step 3. So in step 3, majorly we go with preparing of the flexible residue file. For this, we have to click create macromolecule, choose protein molecule. So wherever you see the choose, better you try to use open as the open option, which will be easier for you to practice. So create macromolecule, open protein molecule. See how I'm going to do. Create, okay, macromolecule, open. So, but you don't have macromolecule. Why? Because it is in PDBQT. Till now, it didn't convert as PDBQT. So, click this all files. Now we can see the latest saved protein. The latest saved protein file name is protein1. So, select this protein1, click open, accept it. Now only it is asking whether you want to save it as PDBQT. Yes, I'm going to save it as PDBQT. Now I am saving a file like protein1.pdbqt. So okay, so now when I am saving it as protein1.pdbqt. Now both the protein and ligand files are in PDBQT format. So I have completed up to this step in step 3. Still we have a part of a step 3, that is defining the active sites. Here for this, select from string. Okay, select from string. Here inside the molecule list, you click here and it can go with the recent state protein, that is protein 1. Then in atom, you have to type in asterisk. The purpose of typing asterisk is the binding site can be any of these amino acids of a protein. So type only one asterisk and ensure that you click only once add. If you click two times add, then it won't work for you. You won't get a result. So click only once. I click only once. This is sufficient. Click dismiss. In order, while practicing Autodoc, you have to precisely do. If you make any step wrong, then you have to close it and you have to delete the folder and files. Again, you have to create a new folder and just place all the files. Okay, so you should be very careful in working with each and every step. So till now, I completed even this step. So the entire third step is completed. This entire third step is completed. Now let me take the fourth step, running AutoGrid calculation. Okay, for this, create set map types. Instead of choose, better use open. Okay, wherever you find choose, use open. Create set map types. Also, you can work with choose, but open is easier for you. Okay, create the set map types. Okay, open ligand. So you have to open only the ligand. Open. Now this step is completed. So after this, grid box. Grid box. So you can able to find a kind of a grid box. See, a grid box is there, a 3D grid box. Okay, now our objective is to enclose the protein in the grid box. I just showed you in the PPT, like to determine the binding sites, you should enclose your protein in a grid box and you can use any of the algorithms. In Discovery Studio Elegant Fit, we are using Fredfield algorithm where we pour water to distinguish the hydrophilic binding sites and hydrophobic binding sites. Now let me right click here. So default you have value as 40. To increase the size to 120, okay, click this 120, click apply. Then only it's getting highlighted now. Now type 120 here, 120, click apply. It is applied. Now right click the green. So to increase the size of 120 here, also click apply here. Okay, and now we can give this 120. So I'm increasing the dimension of y-axis. The next step to increase the dimension of z-axis, click apply, and now you can type this 120. So now you can able to see that our protein is enclosed in this cubical box, but ensure that the entire protein is closed. See, some protein is open where the in the blue box alone it is open. So we have to increase the size. The 120 is not sufficient. Instead of 120, I can give something like 140. Let us check whether 140 works or we have to convert as 150. Is 140. 140, you are perfectly enclosing the entire protein box. If you don't in any sides, don't find the protein is coming outside of the box. So just because we have completed file close saving current, this is the next step we have to make. File close saving current. So we have completed up to this step in fourth step. The next step is to click create output save file as GPF. Create output, okay, save as GPF. When you are saving it, you save like grid1.gpf. Grid1.gpf. Click enter or you just click save. Now this step is completed. That is GPF stands for grid parameter file. Okay, I'm going to say grid parameter file with grid1 name. Now run autoclave launch. So this step takes more time actually after running. After clicking run autocrate, okay, before clicking launch, you have a program path name. In the program path name, click this browse. Okay, we have to select autoclave 4. Click open. See, you can find the entire path here, and also the confirmation changed here the CM. So you should not limit up to that. Okay, we don't have anything log file name. So first click this parameter file name browse, and we have to select this grid1.gpf. If you select this, we are also getting this more URL, like grid log file is going to create. So here you are for creating intermediate files in this step. Okay, so for this, just you have to select the program path name and parameter file name. Obviously, the other URLs are filled. Okay, now you have to simply click launch. So you'll be getting this white window. This white window should not immediately disappear. If white window immediately disappears after completion of that step, then there is some mistake in your procedure of Autodesk. Then what you have to do, you have to once again create a folder, once again download the sample files, once again copy and paste the autodoc4.exe, autograde4.exe, then we can practice that. And before that, before closing, if only if this window getting closed, then only I can take you to this fifth step. Now we have completed all these sub steps in step 4. The next is to go with the step 5 and step 6. Okay, so this alone left out to complete the Autodoc. Okay, but before going to the fifth step, okay, so I know the white window got closed, isn't it? Yes, the white window got closed. So I can go with this fifth step now. What is fifth step? Talking macromolecule set rigid file. Talking macromolecule set rigid file. Macromolecule you have to select protein dot pdb protein. Yes, now that part is completed. So up to this I have completed. Okay. Now I have to docking ligand, wherever you choose, you find the choose, click open. Docking ligand, open, select ligand. Docking ligand, open ligand, you have to do that. Okay, so ligand open. Now it's asking, so whatever window you get, default parameter is to click accept. So accept, you are going to click. Okay, then docking search parameter genetic algorithm. Docking search parameter, GA stands for genetic algorithm. Click this. So here you can able to see two runs. Like if you want to get two best doc courses, you can type 2 instead of 10. There is no need to get 10 docking of ligand, 10 docking process of ligand with the receptor. If you get the one also, that is also okay. But if you want to get two base doc process, select the two runs and click accept. Instead of 10, you are going to type 2 there. Okay, this step is also completed. To reduce the time, I'm making it as 2. Okay, now the next step is uh, 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 docking output Lamarckian. Docking output Lamarckian genetic algorithm. Now we have to save like doc1.tpf. So I'm going to save a file like doc1.doc parameter file. So save this file. So this step is also completed. The next step is run autodoc launch. Run run autodoc, and inside this we have program path name, parameter file name. Like however it did with the autoclave, say here also you have to do the same step. Click browse. Select the autodoc4.exe. Click open. Now similarly click browse. In doc1.dpf we have, isn't it? Click open. Now see entire URL you can able to is filled it in this. Now simply click launch. So you're also getting this white window. If this white window closes, then we can go with the sixth step. Okay. I need nearly 3 to 4 minutes to complete this process. Okay. So meanwhile, I can take you to the demo 6 folder. Earlier we had only 5 files, isn't it? But now see multiple files are there. Doc1.tlg, doc log file, doc parameter file, grid log file, grid parameter file, map files are there. So all these are intermediate files generated in the folder wherever we have created in the desktop. So these files can also be useful for you in future. So I better create a folder so that all the files are also located in the same folder. Got it? I have initially set the path. The initially to have all the intermediate files in this folder. But fortunately, whatever we talk, the file size is not more than 10 MB, I believe, or 10 MB, not more than 100. Okay, the file size is very less. So we can also check what is the. But if you go with this kind of MD simulation, it can also take around 12 GB. Okay, for a single MD simulation, it takes more space. Now it has took around 88 MB, so less than 100 MB. Okay, so still it's executing. Meanwhile, I can show you the sixth step. The sixth step is to analyze dockings, open, and you have to choose the DLG file and analyze macromolecule. You have to click open and analyze conformations load, analyze conformation play rank by energy. So this kind of a tutorial I've already sent to your still the white window not get closed. Already sent to your WhatsApp. So also go through that kind of steps whatever you're practicing. If only if this white window gets closed, then yes, it's closed. Now I can go with the sixth step. Analyze dockings open. Analyze dockings open. So you have to select the DLG file. Click open. So default, click OK. Click OK. The next is actually analyze macromolecule open. Analyze macromolecule open. Even that step is completed. Now come ahead. Analyze conformations load. Analyze conformations load. See, you got two types of energy. One is the best energy. The best energy, how I am telling is negative maximum energy is the best. And if you want to check which is the whether the docking of ligand with the receptor is significant or not, that we have to measure through the value. If we have any docking energy is below -6, then that is a significant docking. The post 2 is -5.39, which is greater than -6. So this is not significant. But post 1 is significant. This is post 1 or conformer 1. Okay, this is the best energy, -7.5 kilocalorie per mole. You can also find these parameters. Okay, so this is how we can make it. So after this, what I have to do, we have to go with the last step. The last step is analyze conformations, play rank by energy. We get like ampersand line, ampersand symbol, click this ampersand. We have build current, click this build current, write complex. Now, instead of PDBQT files, click this all files. Our ligand is actually Indinavir, isn't it? So I'm going to save this Indinavir underscore protease underscore complex dot pdb. So this is the file name I'm going to save. See, ligand protein complex dot pdb in double quotes. If I save this, I have ligand receptor complex in the form of this format. If only if you complete up to this step, then only you can close the window. So don't close the window in between. If you close the window, then you have to do the step from beginning. And because I've completed up to 6 steps, I'm closing this window. Now to analyze the results, what I'm going to do, I'm going to the demo 6 folder. I'm going to this demo 6 folder. Inside the demo 6 folder, I go to this Indinavir protease complex. So I go to this Indinavir protease complex.pdb file, open with Discovery Studio Visualizer. So after opening with Discovery Studio Visualizer, so I can able to get a kind of interaction. See, this is how a ligand is interacting with the receptor. The ligand is this stick model, and this is protein. The protein is the beta sheet. You can find this. If you want to check the hydrogen bond and other kind of interactions, click this receptor ligand interactions. In this receptor ligand interactions, we have showed 2D data. Click this. See, this is how you can find how a ligand is bound with the receptor. The hydrogen bonds are shown in green color. So aspargine 98 is the binding site. And you also find pi alkyl interactions with the ligand. So we have completed how to do the molecular docking using Autodock and how to analyze the results using Discovery Studio Visualizer. And we also discussed on a few methods of computer drug design. Explore more on this. Okay, we can also join the certificate courses which I'll be conducting on December and January. On December, I'll be conducting computer drug and vaccine design, and on January 2026, I'll be conducting AML approaches of drug and vaccine design. Okay, but any doubts you can ask me at present. I have completed my session today. Any doubts? If no doubts, thank you very much for your patience and interaction. So I'll be circulating this video to just watch the video for few times, then you can understand definitely how to make it. Okay, so all these softwares, whatever I provided demo, are freewares, except MATLAB, what I shown you on yesterday. So one can try with any of these tools and databases. Okay, good. Thank you. Have a great day. Bye. Only tomorrow is the last session. Don't forget to attend the session on genomics and proteomics. Okay, and we have 3 more days time. That is 3, 4, 5. Okay, to complete the MCQ. Just merely attending the MCQ is a criteria for getting this certificate, including answering the overall feedback. Okay, thank you. Have a great day. Bye.